Now, during my days as a grad student and as a postdoc, much of my early career at Pitzer was in what Steve Glass refers to as the paleo-terrific days, was dedicated to studying the neural and metabolic underpinnings of appetite and obesity. So how do we regulate energy balance? And what are the mechanisms at our disposal for regulating energy balance? So during one of my grad student revels in the latest volume of Current Contents is now a database, and you simply click on it and it brings you to exactly whatever article you want to read. In those days, you had to read a little white book, and you had to look it up, and then you had to write to the author and hope they would send you back a reprint of their article. None of that happens anymore. And so stuff that would take you a year to get, you can get in seconds now, which is fabulous. But 25 or so, 30 years ago now, I came across a really interesting article by a fellow named Ravelli. And Ravelli was an epidemiologist who was studying the residue, that residue we talked about, of a particular environmental event. And that environmental event was a famine that occurred in the Second World War. So as the Second World War was drawing to a close, in the Nazi-occupied Netherlands, as Allies were beginning to move in, the Nazis imposed a famine. So they put an embargo on incoming foodstuffs into the Netherlands in 1944-45. And that resulted in a famine. And what's unique about that famine is it lasted exactly 6 months, okay? So we know exactly when the famine was imposed, when the embargo was imposed. We know exactly, because of fabulous record keeping, about how much food intake was dropped in those times. And we know exactly when it ended because the Allies came through. You rarely see that kind of window. If you're a developmentalist, this is a really interesting phenomenon, because if you're interested in studying developmental events, you know exactly when an intervention that affected hundreds of thousands of people started, and you know exactly when it ended. And so what you can do, and what Ravelli did, was then to match up those time periods with when women were pregnant relative to that period of famine, and then looked for developmental outcomes. Okay? And what he found was fascinating. So what he found is that if famine started 6 months before pregnancy and ended at the time of pregnancy, really nothing happened. And then he sort of ratcheted his way forward. So if famine begins at about the time of pregnancy and ends at the end of the second trimester, there were a set of effects. And there were two particular effects that were tied to that timing. One, incidence of schizophrenia when the offspring from those women were inducted into the Dutch army 20 years later were much higher. And the second phenomenon was that women who were exposed to the famine during the first 2 trimesters of pregnancy tended to have offspring who were much more obese than the general population. Okay? It didn't happen if the pregnancy started in the 3rd month of the famine, or any other time period. It was only that time period, 1st month through 6th month pregnancy. So really interesting, if you're a developmentalist, that business of what do you carry away with you from early experience? What is the residue? This is a really interesting finding. So I decided as a graduate student to try to replicate that finding. And in fact, if you do this in rats, you undernourish rats during the first 2 trimesters of pregnancy, then you refeed them normally, then you wait for them to grow up. This is the residue. So it's clear that the guy on the right here is carrying much more residue than the guy on the left. These are age-matched, and the only difference between them was that the mother of the guy on the right was exposed to famine during the first 2 trimesters of pregnancy, roughly to the same extent as the people in the Netherlands were during the Second World War. Okay. So if we look at body weight, find is that body weight takes off, especially in males, not so much in females. Interestingly, Ravelli didn't have any data on women because he was using all drafted inductees. But when we look at it in rats, what we see is the effect is primarily in males. Now there are a couple of ways that you can get fat. You can get fat by having more little fat cells, or you can get fat by having bigger fat cells. Okay? Bigger fat cells, there are a number of models that have been looked at in obesity. Bigger fat cells are characteristic of adult onset obesity, and they're also characteristic of obesity associated with a neural problem. Okay, so interesting. And in fact, these guys were getting fat. This is normal rat. These are the guys whose mothers were undernourished, so bigger fat cells. Okay, so what's going on here? We did a whole series of studies looking at what is the material stuff, what's the residue from this early experience. We did a whole bunch of studies looking at metabolism. We looked at a variety of systems. One of the interesting things about timing of this event, first 2 trimesters, and then they're refed in the 3rd trimester, is that this is exactly when critical areas in the brain are differentiating. They're extending out axons, they're forming particular kinds of connections in the brain. So the timing of it is exactly when that happens, and it's exactly when those connections are influenceable by environmental kinds of factors. This is a slide of what's called a neuroblast. It's a cell that will ultimately become a nerve cell, but it isn't entirely differentiated yet. Okay? It can differentiate in a number of ways. It can form many different kinds of brain cells. So this is sitting in a petri dish. The cell is this circular thing, and those white things that you see sticking off of it are called neurites. And it's those neurites that are ultimately going to develop into axons and dendrites, and they're going to enable the nervous system to connect with other parts of the nervous system. Okay? How that happens, precisely what kinds of connections are they going to make, is the real question here. Okay, so this neuroblast is simply sitting in a petri dish with nothing else in the petri dish. The one above it was in a dish with, sorry, the light is a little bright on this one. The one above it is our neurites taken, pictures of neurites taken when the medium in the petri dish had both insulin or had testosterone put into it, which is known to affect neurite proliferation. This one had both insulin and testosterone put in it. Okay, why were we interested in that? Turns out that one of the things that nutrition, undernutrition and refeeding does, is it really affects insulin levels. And the question was, are insulin levels being influenced precisely at a time when they're capable of influencing this kinds of connections from growing neurons? Okay? All right, so we did another experiment, actually there were about 10 between here, where we said, all right, well, if we simply, instead of, so we generated a hypothesis that maybe what's going on with this undernutrition and refeeding is that during the period of undernutrition, insulin levels drop. We know that's true. And it's also true that after a period of famine, insulin levels rebound. And sometimes they rebound to almost 6 or 7 times normal levels of insulin. Okay? Now think about the underfeeding paradigm. During the first 2 trimesters, insulin levels are lowered. Then you begin refeeding mothers. And insulin levels rebound in that 3rd trimester, which is exactly when all of these neurons are sensitive to environmental cues, and they're changing the differentiation patterns. in response to that, okay? So could it be that rebounding from that undernutrition with high levels of insulin is what's responsible? How do you check that out? One way you can do it is to simply avoid the undernutrition and inject them directly with insulin during the third trimester, okay? When you do that, what you find is exactly the same thing happens as when you do undernutrition. So these first two columns here are peak body weight. These are very young animals, so they're just starting to get obese. And you can see that simply injecting them with insulin in the third trimester makes them just as fat as this undernutrition refeeding mechanism. So, we're beginning to get a handle on what's going on here. We also know from 50 years worth of research that there is a particular area of the brain, the hypothalamus, that is very involved in regulating body weight. Okay? And in fact, there is a neurotransmitter chemical in the brain that is deeply implicated in this phenomenon. So we actually decided to measure norepinephrine and found profound differences in both the insulin injected and in normal rats, and in those undernourished refed rats during that third trimester. They're much different than normal rats are. So, okay, so it looks like either insulin or Ravelli's underfeeding, refeeding paradigm are producing the same effects both on body weight and in the underlying neurochemistry. So, cool. That one we can skip. Okay, so this is a picture of that hypothalamic area. So this is what's referred to as the paraventricular nucleus. Paraventricular because it's around the ventricles, the third ventricle. These are norepinephrine fibers coming into the third ventricle. This is a picture of a normal rat. And that's a picture of one of these guys whose mothers were undernourished during the third trimester. If you're a developmental neuroscientist, you stay up dreaming about things like this. So you can hold these slides up and you can tell the difference between these guys. So in essence, you can look at their brains and say, as a result of a very specific in utero condition that I created, I made these guys different. And this is what they're carrying away differently with them. And it causes them to interact differently with the world. They eat more, they don't regulate their body weight well, their metabolism is different. It's that residue that they're carrying with them for the rest of their lives as a function of that early and predictable experience. Okay? It's very cool. Okay. So we published those results probably about 10 years ago. And it's been picked up on, and there are people now who are interested in doing work on genetic models of obesity. So there are, for example, obese animals that are known to have a deficiency of something called leptin. Okay? Now, leptin is a hormone that is produced and released by fat tissue. Okay? And as leptin levels go up, it drops your appetite. Now, that's not surprising if you're thinking about regulation, because one of the ways that you regulate body weight is when you start producing a lot of fat cells, you produce a signal that says, all right, that's enough, stop eating, okay? And it functions very well across all sorts of mammalian species. But there's a breed of rat that is leptin deficient, doesn't produce this hormone. And so what would you predict? So it doesn't produce this hormone that would normally tell you, okay, you've put on enough adipose tissue, stop eating now. What would you predict? They're fat. So these guys are genetically obese precisely because they have not been producing leptin. Okay? So in fact, what we didn't realize, so we knew that was true in adult animals. If you inject them with leptin, they stop eating. But we didn't realize that leptin had a developmental effect. Okay? And if you're a developmentalist, this is great. So this is that same nucleus we were looking at paraventricular nucleus, postnatal day 10, 16, and day 60. This is a normal, WT refers to wild type, so that's just a normal rat. And you can see these neurons in the paraventricular nucleus lighting up in progressive fashion. This is the leptin deficient animal, okay? So not only is there an immediate effect of the hormone affecting appetite, but neurons that subsequently control appetite differentiate differently for the rest of the animal's life. They look at food differently because of that early deficiency of leptin. And what's interesting is that if you replace leptin fairly early on, by day 12, you can recreate exactly a normal character. If you wait longer than that, then you can never make the neurons grow normally again. Okay? So it is simply the lack of leptin that causes an entirely different trajectory in the brain to happen. Okay? All right, so that's sort of where we are up till now. So we've been talking so far about kind of the underpinnings of appetite and the relationship of these kinds of factors to our ability to regulate body weight and appetite. There is a new development, and this is what I want to leave you with because it's really exciting and I hope you will discuss it. There is a new development within the field of epigenetics. Epigenetics refers to ways in which the environment is capable of interacting with the biological substrate to affect outcome patterns. Okay? So often these epigenetic effects influence the expression of a gene. Okay? So it causes different kinds of proteins to be formed. It causes different kinds of neural connections to be formed. So this field is becoming interesting in recent years because some aspects of the relationship between early experience and the later expression of obesity we're beginning to find new cues about that relationship. Molecular biologists are beginning to ask fundamental questions about why clinical rates of obesity are increasing around the world at alarming rates. So in the West, almost 35% of the population is now clinically obese. That's a stunning increase from earlier generations. And although we've identified genetic markers, mutations in the genome that predict obesity, and there are a number of them, both in humans and in animals, we're not seeing a stunning increase in the rates of those mutations, okay? So if you're not seeing an increase in the rates of mutation, how come a lot more people are getting obese, okay? And that question has really led to some interesting epigenetic answers. Okay? There are now a number of researchers looking at these earlier studies, including mine, and are putting forward epigenetic arguments for this phenomenon. This term epigenetics was originally coined by Conrad Waddington in the 30s, and it's used to describe, in general, developmental processes by which the genotype, that sequence of nucleotides in DNA, okay? So there's a sequence of nucleotides that form a blueprint for fingers, for hair color, for everything structural in us, okay? So by which genotype gives rise to phenotype, okay? Our structure, our hair color, that sort of thing. For humans, all of our tissues contain roughly 30,000 genes. Okay? Genes are sections of DNA that code for functional proteins, that produce structure, that produce enzymes. However, in a given tissue and at a given stage in development, only a few of these genes are actually being activated, are actually producing structural proteins that contribute to eye color, to hair color, to enzymes. The real question is, which ones of those 30,000 genes in each of these cells get selected, okay? And until recently, we thought the only explanation for this was random mutation, okay? This is a mutation in the nucleotides, and as a result of that random mutation, you get a different outcome, okay? People who work in epigenetics are now beginning to realize that some of these phenomena that I couldn't explain. So for example, it's not only the case that a mother who is undernourished during the first 2 trimesters produces an offspring who is obese, but that offspring, even if they're treated not normally, will produce an offspring that's obese. And that offspring will produce an offspring that's obese. Similarly, if you undernourish during all of pregnancy, offspring come out underweight. And what we were never able to explain is, even if you nourish that offspring normally during pregnancy, their offspring come out underweight, and their offspring, out to 5 generations. How do you explain that as a genetic phenomenon when the gene isn't changing? There's nothing in the sequence of nucleotides that's changing, and yet subsequent generations are inheriting something. What geneticists don't know how to explain that. It turns out that there are multiple things that can affect gene expression. It's not just the sequence of nucleotides, it's which genes you choose to express. And that's not controlled just by the sequence of nucleotides, it's controlled by how DNA folds on itself. And how DNA is structured in 3 dimensions causes one or more genes to be selected for, to be more likely to be transcribed. Hormones like leptin and insulin are causing genes to configure themselves differently and produce different outcomes independent of the sequence of nucleotides in them. What's being posited in the last 2 or 3 years is that there is a separate mechanism of inheritance that occurs independent of random mutations in the genome, but is influenceable by environmental kinds of influences. This is a revolutionary development, not only in the field of neuroscience, but in biology, because it argues that the experience that one generation undergoes, can be carried out in subsequent generations, and these undernutrition studies suggest out as far as 5 generations, without ever affecting the gene, okay? So this is just beginning. Last 2 or 3 years, this work is just beginning to happen, and it's related to residual effects that influence the way DNA folds on itself and which genes then get selected to be transcribed in the next generation. I'd love to talk to you more about it, but think about that.